Hello, my name is Anna Galelli, and I'm going to do a video that's meant to go with my Pathophysiology 1 class that's going to kind of discuss a little bit about electrolytes and the differences between action potentials in skeletal muscle versus cardiac muscle. All right, so let's focus on neurons and skeletal muscle. So it's not just skeletal muscle. So neurons and skeletal muscles function pretty similarly. This is actually an example from a neuron, which I know because it's got that negative 70 millivolts right here, all right? And this is my resting membrane potential, RMP, which is actually just labeled right here, and this is threshold right here. Now, this is a topic that is covered in your A&P 1 course. Depending on where you are in your cycle of learning, you may or may not have had this information yet. So I'm going to not go into the tremendous amount of details that I would in an A&P 1 lab. This is going to be like a 2 to 3 minute discussion on this. So if you need more details, you will have to go back to the course's list of optional videos and find the more in-depth videos that go with the A&P 1 lab, A&P 1 lecture course. Okay. So first of all, before I get into the electrolytes, I just want to quickly discuss electrical behavior. So this is a graph right here, all right, and it's measuring the electrical energy as it moves through either the neuron or the muscle cell. Resting membrane potential is talking about the plasma membrane or your cell membrane. It has a charge. This is discussed in your A&P 1 lecture. That charge is the negative 70 millivolts. In order for an electrical signal to move, you need to get from here really up to here in order to generate what is called an action potential. All right, an action potential is an electrical signal that goes a long distance. That's a massive generalization. You also have local potentials that behave a little bit differently. Again, you have to go and review the A&P 1 course to get that information. All right, so if enough electrical signal gets to here and it reaches threshold, let me change color right here, it will generate a signal that just goes automatically. It's an all or nothing self-propagated thing, and you will get what is right here, okay? And this is called depolarization when it hits that part, okay? When it starts to go back towards its resting state right here, we call that repolarization, okay? And then it's going to go past threshold, come down to an undershoot where it hyperpolarizes, and then get back up to resting membrane potential. That is basically what this is diagramming here in terms of where the electrical signal is going. Now, what controls whether it depolarizes or not are the electrolytes. Let's look at the next picture. All right, so in our Patho course, we're talking about electrolytes in this unit, okay? And the electrolytes that we're really dealing with in terms of neurons and muscles sending action potential, so action potential, all or nothing electrical signal that can go a long distance. We're dealing, oh, that was the wrong button. We're dealing with sodium ions and potassium ions. Calcium is also involved, but that's a little bit later in this process within the muscle cell, so like if you're doing skeletal muscle, but then it's a lot more involved in cardiac muscle, and we'll talk more about that in a second, okay? So you'll notice that this chart looks really similar. So I've got negative 70 millivolts for my resting membrane potential. I have negative 55 as my threshold potential. I need to get from here to here. So remember, this is describing our plasma membrane charge, okay? In order to get from here to here, I need to bring in more positive ions. In this case, I'm going to bring in more positive sodium ions. Blech, bad handwriting, okay? That is going to make this, which is very negative, less negative. All right, if you put in something positive and you put in a whole bunch of them, it takes more negative to less negative. If it reaches threshold right here, it will do depolarization, okay? Now, once you reach here, you've stopped bringing in sodium ions, All right, now there are going to be these little protein channels in the plasma membrane that open to allow potassium ions to leave. So now you are removing positive ions from inside the cell. That is going to take this, which is in the positive end, and it's going to start making it more and more negative until it reaches threshold. Then it goes past threshold, it goes past it a little too much hyperpolarization. Then we use some other mechanisms and we bring it back up to RMP, or resting membrane potential, okay? And then it's basically at rest again, okay? So this is the role of the electrolytes in generating either a neuron electrical signal or a skeletal muscle electrical signal. And it kind of works similarly in smooth muscle. And this process starts similarly in cardiac muscle, but then it differentiates a little bit. All right, normally I wouldn't give you a picture with so many words on it when I'm doing a lecture, but I'm realizing that a lot of people will not have taken AMP2, and even those who are enrolled in AMP2 probably haven't covered this on how it's different in cardiac muscle, okay? So the first thing we're going to do is kind of talk about this electrical movement of signals, and we're also going to be incorporating those charged ions, those electrolytes, okay? So charged ion, electrolyte, same thing. So we're looking at a very similar graph. All right, so let's dissect this picture really quickly, all right? So here is negative 90 millivolts. That's kind of a typical resting membrane potential for a cardiomyocyte, okay? Now we have in green and red two different lines. The green is really representing the electrical signal, okay? Whereas, so this is the electrical signal. The red is really representing the actual muscle contraction itself. So what the actual muscle cells are doing in terms of shortening and lengthening. So you can see that here they are getting tighter as they contract, and here they are getting looser as they relax, okay? It's not the best explanation, but it's fast. Okay, so similar idea is going to happen as what we were looking at with skeletal muscle, okay? So when you start out, it's very similar to skeletal muscle. So this is step one. We are allowing sodium ions to come in, all right? And that's going to start taking it from very negative to less negative, all right? And eventually it's going to reach a threshold, and then it's going to generate an action potential, okay? And that's kind of what they are describing right here, okay? Now, as it is doing this, I've got, what's different here is I've got calcium ions that are going to start entering the plasma membrane, all right? Not a lot, but these are extra. I'm going to just abbreviate this, ECF, extracellular fluid calcium ions. Okay, so this is your electrolyte. These are in the fluid outside the cell. Gates are going to open that allow this calcium to come in, okay? That calcium is going to go to a structure in the sarcomeres that it opens, okay? And that's basically kind of what it's describing right in here. So it opens up these gates, which allow a ton of calcium that is in, I'm going to abbreviate this SR, sarcoplasmic reticulum. That is the same thing as endoplasmic reticulum, except for inside the muscle cell. So it's going to release a ton of this calcium. This calcium, I mean, it is so much right in here that it creates what is called a plateau period, where there is so much calcium that it maintains the generation of the action potential, which in turn maintains the muscle contraction. Okay? So you basically have these two waves of calcium electrolytes, calcium ions that are going to come in and maintain this. Now, why this is important is I want you to think about one, let's think about absolute. I'm going to just write this absolute refractory period. Actually, let's use something that might be simpler if you haven't done A&P1. Let's do how long the contraction lasts. All right, so how long the contraction lasts. I've got skeletal muscle and neurons. All right, they do not use this calcium, actually, let me erase that. They do use calcium, so I don't want to confuse you, but they do not use, so no extracellular fluid calcium, okay? That means their contraction is a short length of time, okay? It lasts about 2 milliseconds. In contrast, cardiac muscle, all right, it uses extracellular calcium for first wave and then tons of intracellular calcium to keep it long. All right, and in fact, it's so long, we are talking about 200 to 250 milliseconds. Okay, so this is a huge difference compared to skeletal and to neurons. Okay, and this is important because we're trying to get that entire chamber that has blood inside of it to tighten up all at once and to maintain pressure long enough that the pressure increases and gets high enough to open up the valves so that the blood moves from high pressure to low pressure. Okay? All right, now let's look at this next picture, which is the one that was used in your Patho PowerPoint. So this picture is showing you the same thing that I showed you on the previous slide, but it doesn't show you the muscle relaxation in relationship to that plateau. So this is representing depolarization and the influx of sodium. This is your plateau period due to the influx of potassium ions. Now, not potassium ions, calcium ions, calcium ions. Now, potassium is going to leave and you have the repolarization period where you're going to get back to the resting membrane potential. Okay? All right, now I don't want you to get this confused with things like the EKG. Okay, so y'all have probably all seen pictures of EKGs or ECGs, where you've got the little blip, or you see like TV shows where they've got the monitor from the hospital room and you're seeing it. The two pictures I just showed you have nothing to do with that. Okay, they do represent the electrical signal, but they're representing it in a different way because they're focusing on the influx of the electrolytes, not the actual movement of the charge across the heart. All right, so let's just look at that really quickly. All right, so this is an ECG or EKG if you're feeling German, okay? So yes, the previous diagrams can show this, but what this is doing is really showing you the movement of the electrical signal in different views on the chart. And they correspond to the different chambers. So the atria depolarizing or contracting, the ventricles depolarizing or contracting, and then the ventricles relaxing, okay? But what they do not show you is the movement of the electrolytes, okay? So that's what the previous diagram was showing you. All right, I think I can end this right here. All right, I think this is enough of an overview that we can get you through those two relevant learning objectives. All right, so thank you for coming, and go take a break.